Good afternoon, everybody. I'd like to welcome you all to a special co-hosted and co-sponsored webinar brought to you by the Lake States Fire Science Consortium and the Tallgrass Prairie and Oak Savannah Fire Science Consortium. I'm Jack McGowan-Stinsky, and I serve as the program manager for the Lake States Consortium.
Hi, everyone. I'm Craig Meyer. I'm the coordinator for the Tallgrass Prairie and Oak Savannah Fire Science Consortium.
Both Craig and I will be moderating the webinar this afternoon. Today we have a webinar that highlights microbial community responses to fire in Remnant and restored grasslands. This is also part of our Manager Scientist Lessons Learned series. For those who may be joining us for the first time, let's give you just a little background. Both the consortia are funded by the Joint Fire Science Program and are two of the 14 regional consortia across the country. Our mission is to accelerate the awareness, understanding, and adoption of wildland fire science information by federal, tribal, state, local, and private stakeholders. For the Lake States region, this includes Michigan, Minnesota, and Wisconsin, and the adjacent Canadian provinces of Ontario and Manitoba.
The Tallgrass Prairie and Oak Savannah Consortium covers portions of 11 states, encompasses much of the historic range of the Tallgrass Prairie, reaching from southern Michigan and central Ohio to the eastern Dakotas and Kansas. We're working with a wide range of people with a stake in managing fire-adapted ecosystems, from private landowners to nonprofit organizations and state and federal agencies, as well as researchers. The consortia strive to be inclusive and neutral science partners, so we work to foster collaboration among researchers and practitioners, organizations, and individuals. We facilitate dialogue about new science findings and emerging needs and develop innovative approaches to science delivery.
Before we get started, I want to take a few seconds and explain our webinar interface using Adobe Connect. First, if you would like to ask a question or interact with the attendees, please use the chat box located in the lower left-hand corner of your screen. Once you type your question, just click the send button. Craig and I will be monitoring these questions and will make sure that there is an opportunity to address them. Second, we recommend that you maximize the webinar window on your computer.
Finally, if you'd like to learn more about the two consortia and what we're up to, please visit our websites. You can look for information at www.lakestatesfirescience.net or www.tposfirescience.org. Please sign up to receive our newsletters and announcements of other activities, which you can find links to that on our homepages, as well as our Twitter and Facebook links are found at the bottom of the presentation as well.
All right, today we have three speakers, Kathryn Docherty of Western Michigan University and Ryan Koziatek and Ashley Wick of Kalamazoo Nature Center. I'm going to let them introduce themselves as they start to talk. Okay, guys, the webinar is all yours.
Thank you, Jack. All right, so I think this is the first time for three of us doing a three-person presentation, so we'll try to keep it as concise and clear for you all as we're going through. My name is Ryan Koziatek, Stewardship Field Director for the Kalamazoo Nature Center. Basically, I look over the land management for the KNC grounds, but also manage a seasonal 5 to 6 person field crew throughout the year. And on this side, I'm also on the Michigan Prescribed Fire Council as the Outreach and Education Chair.
Hi everyone, I'm Kathryn Docherty. I'm an assistant professor at Western Michigan University, also located in Kalamazoo. And I've recently begun a collaboration with Ryan and Ashley to examine what's going on below ground with the microbial community in a restored prairie. And this stems from an interest that I have in understanding how microbial communities are impacted by human activities as well as by climate change, and some of our studies are moving in that direction.
Yeah, hi, and I'm Ashley Wick, also of Kalamazoo, but calling in from Chicago, and I'm really excited to talk to everyone about our new collaboration, and excited for us who usually only look at above ground aspects of prairie restoration and to start using this new partnership with Kathryn to look at the below ground as well.
All right, well, as we get through this presentation, like I said, there'll be three of us going. I'll be speaking first, followed by Kathryn, and then Ashley will be kind of wrapping it up. The main point we're kind of getting across is those lessons learned within this collaboration. So in between those are going to be a little bit of the meat of what I do, what Asha does, and the meat of the presentation with the microbial communities. So prepared to get 3 perspectives. As I said, I'm a land manager for the Kalamazoo Nature Center. Kathryn's academic researcher working with Western Michigan University, and Ashley is the research director at the Kalamazoo Nature Center. A little bit about the Kalamazoo Nature Center to give you a little bit of place. You can see the 2 consortia on the right maps. That red star is where we are located. So we're in the Tallgrass Prairie Oak Savanna Consortium area, but we're also on the very edge of the Lake States. In addition, what we'll be talking about today, those microbial communities, is something that might be interesting to look at, and not just the region that we're checking out. So the Kalamazoo Nature Center was established in 1960 by some professors at Kalamazoo College here in Kalamazoo area. We are focused on education, development, and conservation stewardship. That conservation stewardship department is what Ashley and I are a part of. And the conservation stewardship department, we do everything from ecological services to surveys. We've got some large and small clients that we do restoration for as well. On site, we have about 1,100 acres that we look over with a diverse habitat. The spot we'll be placing you today is actually off the Kalamazoo Nature Center grounds, a place called Harris Prairie in Oshimo Township. It's just west of the Kalamazoo Nature Center by a few miles. Your map on the left here shows our study area. That area in blue is the Remnant Prairie. The area in beige is a planting that was done under CRP, or Conservation Reserve Program, in 1993. The 2 orange squares are 2010 planting, also with a Conservation Reserve Program money, but a different seed mix. That 1993 seed mix was pretty early in when people were planting grasslands, and so it's very high concentration on warm season grasses and not so many forbs. So we've got more diverse community on those outer squares. And then just to the east, that's not highlighted, that other field is a, because of this collaboration, we've moved up, and it's made it possible for us to do a new planting here, and then we'll talk about a little bit more. From the history of land management for Kalamazoo Nature Center, we've been pretty involved from the beginning, although on a small scale. So in the early 70s, we had some prairie enthusiasts like Bob Plesnick that were doing prairie plantings and maintenance in the early 70s. 1993 was that planting that I had mentioned. 1994, there was a land use plan for the Nature Center, not intended as a land management plan. It slightly mentioned stuff about habitat management. So it wasn't until 2003, 2004 that some intensive bioinventories were done. In 2005, finally had a land management plan. Now we focus mainly on invasive species management, restoration, habitat restoration, and then fire and other tools for maintenance of different habitats. While we're going through and working on projects, whether it be invasives or fire, we like to use adaptive management. But there's been one part that has kind of been lacking a little bit due to time in other areas, and that's that number 3, and that's monitoring. It never seems enough time or planning in for that, or for some really meaningful metrics. So how do we know whether our land management is working? I mean, that's one of the things that I'm pretty concerned about when we're putting time and resources and people's health in the way of getting some of these habitats managed. So, you know, not enough resources for monitoring, or even having those meaningful metrics or protocols that could be used not just on our site for what would work there, but outside of that as well. And of course, I mentioned time here too. On the right, you can see a picture of our field crew. There's 5 to 6 of those every summer. And then I also run our burn program at the Kalamazoo Nature Center too, which deals with training and equipment maintenance and all that as well. So as most of you know, life of a land manager is long in the hours. So had all those shortcomings and limitations as a land manager, Ashley and Kathryn had approached me. They had been talking about working in one of our prairies, that Harris Prairie that I was showing you. And they had some technical questions for me in regards to burning, in regards to putting study plots within the units, and you know, how they could do that. You can see in the photo there, just along the left-hand side, we had to actually put one of our burn breaks right through the middle of the unit. So that, you know, that has a couple obstacles there, not only with the fire, but also wanting to maintain the entire area. But when they started talking a little bit more about the microbial community studies that they wanted to do, it was something I had considered kind of on a whim. Maybe have grabbed soil from my remnants and say, oh, I wonder what this would do here. And that's about as far as it got. So when I had the chance to get even a little more involved, it was very exciting. So started to work a little more with them. So I'm going to turn it over to Kathryn now, and she's going to talk to you just about what we were looking at.
Thanks, Ryan, for that great introduction to the sites that Kalamazoo Nature Center has been kind enough to allow me to work in. As Ryan was mentioning, land managers tend to use above ground metrics for determining whether and how long a restoration has been in place and whether that restoration is successful. So some of the things that I've learned from Ryan and Ashley is that land managers tend to look at biodiversity. They use the floristic quality assessment metric. They might look at the presence of indicator species, such as lupin, to see how well restored an ecosystem is becoming. And they may also look at things like recovery of above ground ecosystem services, like the presence of pollinators and other types of predators. What I'd like to talk about today is something that we don't often think about. What's going on in the invisible area below ground in the soil, below our restored and remnant prairies. So just a little soil taxonomy 101. We have pretty highly fertile soil in Michigan that has a loose organic matter layer and a mineral matter layer and humus directly below it. All plant roots are going to be found mostly in these two upper layers. And most microbial activity, which is what I am interested in, will be occurring in the top 7 centimeters of the soil, or that loose organic matter layer. So all samples that we're looking at here to look at microbial communities below these prairies are coming from that top 7 centimeters in the organic matter layer. So as I mentioned, most plant roots are going to be in those top 2 layers. And we know of at least 4 different ways that agriculture, or historical agriculture, can have influenced soil microbial communities. The first I want to talk about is the impact on arbuscular mycorrhizal fungi. So what are arbuscular mycorrhizal fungi? They are fungi that interact with plant roots and create hyphae that extend the plant root system and cause an interconnectedness between plant roots. This is a beneficial symbiosis for the plants that lie above them. The hyphae allow nutrients and water to be transported to the above ground plant biomass. And the above ground plants are transporting carbon and sugars down to the arbuscular mycorrhizal fungi. So this is a relationship where both organisms benefit. And what we know is that particularly extensive tilling that's involved in agricultural planting really disrupts this mycorrhizae plant root interaction. And mycorrhizae are obviously storing carbon in this system. If they are taking carbons and sugars from the above ground biomass, they are a significant storage of carbon in this below ground top 7 centimeters. If that's disrupted, then this prairie may not be storing as much carbon as one that has never been tilled. Okay, so that's the first I want to talk about, and that is one thing that we're going to be looking at in the terrace prairie system is how many arbuscular mycorrhizal fungi are present in each of the dated restorations that Ryan mentioned. What we did in September, right before and after a burn, was to look at just the 1993 restored prairie and the remnant prairie that's not been agriculturally cultivated. And this is just a quick comparison of the soil nitrogen levels that are present in each of these two prairies. So we know that fertilization causes a really long-term impact on the amount of soil nitrogen that remains in a system. And what we're showing here is on the left, ammonium, and on the right, nitrate is much higher in the restored prairie still after 21 years of initial restoration than in the remnant. So there is this long-term impact of prior fertilization that is still ongoing with this nitrogen fertilization. Well, what is that doing to our below-ground microorganisms? It actually has a really significant effect. So we talked before about fungi. There's another group of organisms, bacteria, living in the soil that are performing many different biogeochemical cycles that are important to all ecosystems. In a never fertilized system, what we tend to find, and what a really groundbreaking paper just published last year, is that there are more of this phylum Microbia, a group of bacteria called Microbia, that are really slow growing and that use really large pieces of carbon instead of small, really easy to use pieces of carbon. So recalcitrant carbon is this really large carbon that is difficult to break down. So this type of a system is generally termed oligotrophic. It's something that is a slow-growing and very low nitrogen system where recalcitrant carbon is what's dominantly used. In fertilized systems, we see something completely different. We tend to see more of the groups actinobacteria and firmicutes, which tend to be much more labile carbon degrading organisms and are adapted to high nitrogen amounts. So these are termed copiotrophic. They are sort of the weedy species. You can consider them as more weedy species that are growing in these high nutrient environments and that they may grow much more quickly. The ultimate result of this is that these two communities represent alternative stable states. So an agricultural field, once it's planted, once it's had tons of nitrogen put on it, is going to have this effect of past fertilization for an extremely long time, even after restoration. So what's the point of that then, is that in our prairies, we likely see much less carbon storage in restored prairies than in remnants. And that's one of the things we're testing with the study that we did in September right before the burn, is we have this assay where you can measure the amount of carbon utilization using a 96 well plate assay that measures 31 different carbon substrate uses at once. And basically what we see is that on this graph, this is just a measure of carbon substrate utilization. The restored prairie microbial communities are using labile carbon really much more quickly than in the remnant prairie. So they are cycling carbon more quickly in the restored prairie. What does all that mean? If you take it all together, we have less arbuscular mycorrhizal fungi, more nitrogen, different bacterial communities with different carbon utilization strategies. The overall take-home point is that it's likely that in a restored prairie, there is a lot less below-ground carbon storage than in a remnant. And we know this to be true from a study that was published in 2006, where they examined a prairie that was restored 7 decades ago, and it still had very different carbon storage and carbon budget than an adjacent remnant. So that led us to wonder what's occurring following a prescribed burn with these microbial communities, especially immediately after a prescribed burn. Does prescribed burning have the same beneficial impact to restoration on microbial communities as it does above ground? Well, what we know from the prescribed burn that Ryan coordinated in September, and then where we sampled a month later as well, so we sampled the day after a fire as well as 1 month later, what we know is that soil pH changes. It becomes more basic immediately after a fire. Also, nitrate and ammonium levels increase immediately after the fire. And if we go back to that carbon utilization assay and look at how well bacteria are using different amounts of, or different types of very labile carbon substrates, we see that it's mostly this impact of the restored versus remnant that's driving the response to fire, and not necessarily the inputs of above ground carbon inputs that the fire may have had. What we do see is a trend of immediately after the fire. In the restored prairie, we see an increase in carbon utilization. Not a significant trend, but it does occur. Whereas in the remnant, there is a trend of decreased carbon utilization immediately after the fire, within 24 hours. So I thought to explore that just a little bit further, I'd look at just the metabolism of nitrogen-containing carbon substrates in each of these samples. And what we see is something really interesting, is that in the, again, using that biolog assay that is measuring carbon utilization, this is just an amino acid asparagine that I'm showing that contains nitrogen, we have much more asparagine utilization immediately after the fire in the restored than in the remnant. So these microbes that are in the soil are going after carbon substrates that contain nitrogen because they're adapted to really high amounts of nitrogen. So they probably are adapted and have enzymes that can break down carbon substrates that can contain nitrogen much more than microbial communities in the remnant prairie. So the take-home point is that we have really different microbial communities in restored and prairie soils that fundamentally perform different ecosystem functions. So when we look at above ground, whether a system is restored, that may not translate into increased carbon storage or increased ecosystem restoration. Below ground restoration tactics are something that Ryan and I and Ashley and I have been working on thinking about in terms of how can we add in some sort of a below ground component that fits within these constraints that Ryan mentioned about land managers needing time and resources to perform these types of tactics. And so going forward with our collaboration, that is where we are starting to take our work and where hopefully we'll have some interesting results in the future to present again. I think Ashley is going to then now talk about the overall collaboration between land managers and academic researchers and a little bit more about our future directions.
Yeah, great. Thanks, Kathryn. And thanks, Ryan, too. And I would like to point out how incredibly lucky I feel to be working with two people that can so thoroughly describe, our site and what our goals are there, and also the methodology. So when Ryan proposed that we do this webinar with the three of us combined, I said, well, Ryan, you talk about the site, and Kathryn, you talk about our methods, and you guys don't need me. And they brought up that my position as research director at the Kalamazoo Nature Center is quite unique in that I kind of have to wear both hats. Sometimes I have to be a land manager and work with Ryan, and sometimes I'm a researcher, and I am primarily a conservation biologist. So since we could have put wearing both pants because of the day of our burn in September, I went from sampling from, I think, 6 a.m. until noon to lighting the fire and putting on my PPE at noon and being out there until the end of the day. So a few things about the type of position I have at the Nature Center. Primarily, I work with a lot of researchers, and one of the things I do is grant permission to those who want to do work on our properties. And so I actually first met Kathryn when I was taking her on a site visit, and since then our project has evolved, and I really think it's going to be quite meaningful to have these results and methodologies that we're working on that will also be really useful to practitioners like Ryan and I. I also run a statewide initiative, the Michigan Butterfly Network, which is actually the reason I'm in Chicago for a national meeting right now, and do some climate change work in Kalamazoo and a few other projects, and mostly butterfly-based. So what I wanted to talk about, just a little bit about our project, is that, we all have limitations, and I think that that's why we're all so happy to be working together. So for instance, with Ryan and myself, when I have my land manager hat on, managing the land is a full-time job, and like Ryan said, he is managing contracts from other organizations and institutions, managing our field crew, and a pretty large amount of budgeting he has to do to make that all work throughout the year. And also, when you're a land manager, you're by nature geographically limited to the land you're managing. And land managers tend to have more of a holistic view of the place they work rather than having very specific questions. Or they may have those questions but don't have the time to answer them. Whereas researchers tend to use many systems and are often kind of spread out geographically, working on several different projects. And they tend to be more narrow in their hypotheses, because we all know that when we're trying to get funding, we do need to have these very specific questions we're asking. And then when you compare land managers and researchers, I think that we often find that land managers tend to stick around the same piece of land they manage for, decades or even careers, whereas researchers often have to, move on with the trends of what's getting funded. And so we often have, someone that will do a PhD on a parcel of land, and their project will be, 2 to 5 field seasons, and then that knowledge may be lost and may not be in a publication that a land manager may think to look. And then, of course, we're all limited in certain ways. So if we had the time, we would be managing our land to get rid of all of our invasive species and making sure we're able to burn all of the parcels that we have targeted, but it just does not happen. And also in Harris Prairie, we have to be aware of some at-risk species we have there. And not the Kerner blue, as the picture of lupin might suggest. And then, of course, in the end, we're all limited by funding. And so that last limitation is something I want to talk about, that, in particular, all these limitations are something that we're dealing with this collaboration, and we're actually benefiting. So with the funding, we're taking actions such as prescribed burns in very specific places that Kathryn and I need to have done for our research, and that Ryan has in his land management plan, and we're making it so that it benefits everyone. And it does take, a couple of, Ryan maybe making some concessions for us, and us maybe making some concessions in study design as well. And then, so this is actually my last slide, and then I'm going to open it up for discussion with Ryan, Kathryn, and I to talk about how our work together has gone so far. I think it's been about 6 or 7 months. But I would have to say that so far as learning from each other, we've learned that we have to have really open communication, and it's pretty common sense, but it really does mean that we have to talk about even the smallest concerns we have. And Kathryn and Ryan and I have been sitting down for very long meetings as we were getting ready to submit a pretty big research proposal. And these hypothesis brainstorming sessions have actually been incredibly useful for, I think, everyone involved, especially Kathryn, who's had to do the majority of the writing of these proposals. And we started a literature reading group before these meetings, where we would all pick 1 or 2 articles that were relevant to the work we're doing, and then have a chance to discuss them. And I know that, in particular, from the land manager's point of view, is that we don't always make time to read the primary literature. It's incredibly vast, and it can be overwhelming. And to sit down and talk about it and read studies that have been really relevant to our system has really helped us, I think, all 3 of us gain perspective, especially as we talk about potentially developing new methodologies. And that's my last point. And yeah, I think it's been really important to have Ryan involved with big research ideas and these new ideas, that these methodologies need to be cost-effective, they need to be doable. So to have us all in the pre-proposal stage really talking about the usefulness of what we're going to be doing. And then back to, from Kathryn and I's point of view, with getting funding, I think that going through all of this work of primary research and knowing that it actually is needed and will be useful to practitioners is huge for us, and hopefully Ryan agrees with us. So with that, I just want to bring Ryan and Kathryn back in, and you guys we give you a picture to look at while we chit-chat of a GPS unit that got left in the field by a field assistant of ours. So Ryan, if you want to get started.
Yeah, certainly. So one of the things you touched on briefly, and I can give an example of, are some of the concessions we have to make back and forth when we're putting our goals out on the table. So in the design for actually saving aside some for baseline studies, we had to put our control plots in, and so these are areas that not only did I not get to burn this year, but I may not get to burn until at least the project's over for the time being. And we've been trying really hard not to stay in those nice little square lines that have been set up for us politically. So, you know, when we're going towards managing into those ecotones, it's one step back I have to take. But again, I was so, eager to get involved with the project, but so there's a couple things that I'm okay with actually doing. So mowing that burn break through, at 90% of the field, so 10% of it is left as control. I can work with that for a few years, and then come up with some other techniques, especially in the prairies, to take care of woodies while we're out there.
Mm-hmm. Yeah, that's a great point, Ryan. I think that also we have to sessions, but establishing control plots all in one line instead of the remnant or the restored prairie, was one thing we had to do, so that burn break could be mowed along an edge of the prairie, and then all the control plots are in that edge. So that's a issue we can deal with statistically, but it's better than having to mow burn breaks around tiny little plots for each of the controls and then try to protect them from a fire. Especially when you have 10, I don't think we have the people to do that.
Right. Ashley, can you think of anything in there for hurdles?
Well, I guess it doesn't necessarily fall under the hurdles, but as I hear you two talking about this, it makes me think about the point that Kathryn and I are at in this research project, in that we are not yet funded, and we are collecting all of this data for preliminary data to eventually get funded, right? So our ability to find a partnership with Ryan, who is willing to, tailor his burning season to match our needs, was just so important, and I'm really thankful that he was able to, see the benefit in the long run for him, even though it may take, a bit of extra time at this point. I guess that doesn't fall under the hurdles part. But I don't know, we're making it all seem so nice. Can you guys think of any other hurdles we've had to jump over?
Well, one of the issues of this fall burn that we did was, you want to talk about that now?
Go for it.
Was that the conditions have to be right for burning, as you all know, and that the conditions happen to be right much earlier than we kind of had anticipated for Ryan. So,
Ryan, do you want to talk a bit about our burn timing this year? Yeah. Or I should say last year.
Yeah, no problem. So the actual burn ended up being on the 25th of September, and it was probably a week before that Ashley approached me and says, hey, do you think there'll be any burning in the next couple of weeks? And I said, it doesn't look likely. Well, that didn't seem to be the case. Just 5 days or so later. And in fact, I had left them given even a longer time window, where it would maybe not be till mid-October to early November, depending on, of course, the conditions. Well, it was those conditions that snuck up on us. So if you look at your map, you can see US 131 right to the east. For those of you not familiar with that area, it's a 4-lane highway, and we're only about a quarter-mile from that. So big issues with smoke if we get winds coming out of the west. So we had great drying time, which is nice. In that 1993 planting, I mentioned it was very warm season, grass heavy. So I've been looking to try to do a growing season burn, or at least late summer, early fall for that. So we had enough drying time for that, for the underground stuff, and the fire had to go. So I gave Ashley 24 hours notice just about, and then she connected with Kathryn.
Yeah, so Ashley, immediately said, we got to go in the field tomorrow morning and collect samples from the control and experimental plots in the Remnant that we hadn't collected pre-fire yet. So yeah, this is the day that she mentioned that we were out at 6 in the morning collecting samples, and we collected samples from all the experimental plots before the fire, and then Ryan lit the fire, and we were in the control plots collecting samples while being very carefully protected by Ryan and his crew while we were sampling. So we're still sampling the pre-fire control plots. And it really came down to having to just understanding that that's maybe what's going to happen in terms of working with land managers who are trying to manage land within the constraints that they have and with the weather constraints that they also are dealing with. So being prepared ahead of time is something really important. So maybe if you think a fire is going to happen in 2 weeks, be prepared that it might happen tomorrow. I used to work in a system in California, and I was told the day ahead of time that I had to fly out right before a prescribed burn to get samples. So it's far more convenient to have to drive about 5 miles down the street to get these samples. So it is an important consideration, though, is that it may not happen for the academic researcher in the time frame that we want it to happen, but you still have to be ready to mobilize as soon as possible.
Yeah, and actually, probably one thing we may do better later, we haven't discussed it yet, is when we're thinking about planning, when I'm doing my burn plan, including Kathryn in that planning process. And also with her project set up too. I mean, I heard a little bit of it, but I wasn't as eager to be involved with her side of the planning as I was on my end. So I did consider that they would be out there with the burn plan, and made our adjustments for that, and of course included them in on our briefing that day when we were doing the burn. But yeah, that was one thing logistically was kind of a hurdle, but we were able to mitigate for all that.
Yeah. And I just want to use this as an opportunity to point out that some of the data Kathryn and I have been collecting is pretty cool to look at. So aside from huge bags of above ground biomass, which you know how late season big blue stem gets, we also, right before this fire, had, so the night before the fire, Kathryn and I had stayed up quite late painting metal rods with the paint that changes as it melts. Kathryn, what increments are those in, the temperatures?
Oh, I think it's every 10 degrees Fahrenheit. Or
yeah, so
like
you know, just as far as interesting intel we can offer Ryan, who, you know, I think we're all kind of curious, is like, how hot is our fire getting out there? So, we're able to share some of that anecdotal data with Ryan, and yeah, so things like that have been kind of cool. You know, how hot is our fire burning this time of year? So.
Yeah, and above ground loads of biomass also, I think it could be an important thing for the future, is knowing exactly how much biomass is out there, which we now know. That was another hurdle. I didn't quite realize that the tall grass prairie would take up an entire greenhouse in terms of drying above ground biomass. So we learned from that after the first sampling. But yeah, now we have that data that can go forward in the future and hopefully inform how much biomass is there to burn.
Mm-hmm. Another thing I appreciate about this collaboration too is, some of these methods for collecting data are, stuff that is either been developed by Kathryn and Ashley based on the local conditions, or they might be something that's, out there, and implementation might not be that difficult, but just getting those kind of protocols and having that knowledge ahead of time to get that data collected is opened my eyes a little bit to how we can look at some of our other properties.
Yeah, and so soil coring takes muscles, so. All right, well, I'm going to move on. Do you guys have any last minute comments on that part?
Guys, I'll jump in. There is a question, and it's which one of you initiated the collaboration, and how did you find one another?
Yeah, I think I'll take that question. So I actually only started at the Nature Center last March, and one of the first things I had was a voicemail from my boss telling me that part of my job is to arrange site visits with potential researchers and make sure that whatever work they're doing on our property isn't going to be too invasive, and that it's in the right places, and helping someone, especially Kathryn, who's only been in Michigan for a few years, help them find the place that's right for their system. So Kathryn and I actually met on that initial site visit. I took her to Harris Prairie, and I don't know, Kathryn, was it kind of a couple months later that we started talking about data sharing arrangements, and then from there I think we started talking about the questions you were asking, and I started to get interested, and I think we started looking at it a bit of a different way, if that's correct. I'll let you jump in.
Yeah, definitely. Yeah, it was a good opportunity. I was talking to some of my colleagues here about where I might be able to set up plots that I could put heat lamps over to do a warming experiment for a proposal I was writing for the Department of Energy. And they took me, Ashley and her boss took me out to Harris Prairie, and we were looking at Harris Prairie and started talking about restoration, and I hadn't really considered restoration as part of what the project might entail. And then I think one of the main things that I really like about our collaboration is that we've started a reading group and talk about papers as well as different land management strategies, and Ashley's been more than generous in taking me out to many sites around town and showing me different prairies that we might be able to work in. And I think that through that I've become much more interested in restoration and understanding what's going on below ground, mainly because it gives me some sort of a very tangible impact of my research on what land managers might be able to do in the real world. A lot of times with warming experiments or and many scientific experiments, you don't get that tangible feeling that your research is going to actually change something. And in this case, I really do feel that way, and it's definitely an exciting feeling to have.
Hey, Kathryn, did you have any conversations with anybody else at Western? Because we've done some different work with other professors, so I don't know if anyone suggested looking at the Nature Center as a potential site.
Yeah, definitely. There are a few other researchers who have at least talked to folks at the Nature Center about doing research. I'd spoken to Steve Kohler and to Sharon Gill and Martin Von Hoff, and they had recommended talking to Nature Center, Southwest Michigan Land Conservancy for these projects, and that's how it led me to Sarah and then Ashley.
Yeah, the reason why I asked that because we've been working as a partner with Western on, you saw a litany of the stuff that Ashley's looking over as well. So there's relationships built elsewhere other than this project, and it was through those, some of those doors that had been opened that some of these eyes have come through. So it wasn't completely serendipitous, but there was definitely some work. One thing we're talking about with a Kalamazoo College professor is how we can get interns into the field with some meaningful experiences, and just basic RFPs that we can, or requests for proposals that we can send each other saying, I have these students, or we have these areas available for study, and then those are out there for somebody to browse if they needed.
Yeah, and I will just wrap up with the answer to that question we had, is that I've been working in restoration of tallgrass prairies for a while now, and for me, when the idea of looking below ground and finally finding someone that has all that huge set of expertise, with restoration, I think that it has been so important, and I've always been curious about how to look below what we're always seeing. When we say, oh, our restoration is successful, we have some beautiful forbs, we have tall grasses, and we've put fire on the landscape. And how do we really know when these restorations that are looking pretty good are actually, are they ever getting back to some sort of baseline? And what we're seeing is, maybe no, maybe they're just, like Kathryn said, reaching this alternate stable state or steady state. And so the next thing that we're really excited at looking at is looking at restoring microbial communities as well as that, above ground structure that we all want to see in our prairie restorations.
Yeah, that's a great point, Ashley. And one other thing that has crossed my mind is that what if we can come up with a least common denominator metric that we can measure as a monitoring aspect to determine whether below ground restoration is proceeding as we'd like it to. And as Ryan mentioned, there's not much in the way of resources and time for monitoring, but if we can do some very simple assay to show that below ground we're starting to look more like a remnant, I think that might be something that's useful. So that's one of the areas that I want to start thinking about next is what is the least common thing that we can measure and we're getting at a more restored below ground community.
Yeah. So I'm going to go ahead and do a wrap-up slide, and then we'll take any additional questions. So thanks, Ryan and Kathryn, for that discussion. So I just wanted to kind of sum up and say, we got this first season of preliminary data, and it was a lot of hard work, and I think we're pretty confident it'll pay off. We're also really happy with our new relationships. And also, I think that the 2015 planting, the cornfield that was just between our sites and the 131 highway, we're planning on planting that next year, and we kind of got moving and taking one of those last parcels of Nature Center property out of lease to a farmer. So we're pretty excited about that. And one last thing is that as part of the proposals that we're working on, we're also having a community outreach portion where we're going to try to offer some sort of class to, and our target audience is going to be individuals who are landowners and helping educate them on restoration of their own lands and the services available and what they can do themselves. And with that, I'd like to thank everyone who listened, and we'll take questions.
Okay. There are a number of questions. I'll try to take them in the order that they came in. One's a specific or more specific one. It's asking, after getting rid of the brome grass, they have a 20-plus acre restoration that they're working on, and they're contemplating an application of compost or other. Looks like they're looking for advice, maybe to boost the microbial community.
Does the compost have a lot of nitrogen in it? I can't answer it because I don't know what the community composition of the compost would be, but I would imagine if you're putting something that has compost in it, you might be putting in a lot of decaying and nutrient-rich organic material, which may actually have more of a fertilization effect.
Okay. And I guess kind of more general with that, the question that comes to mind is, do you have suggestions for, in these remnant prairies, kind of boosting the microbial community or alternate or changing it back?
So what was the second half of the question? Sorry, Jack.
So if you're comparing a remnant versus a restored or a restoration project, is there a way to kind of shift the microbial community back so your restored prairie starts looking more like the remnant?
Well, we're thinking of, that's a great question, and that's what we just submitted a proposal to attempt to do, is see if we can take a remnant type microbial community or remnant soils and determine what's the most efficient and most successful way of establishing that into a restored prairie.
I think, I mean, when in regards to is fire doing that, I think there's much more happening below ground, and there's thousands and thousands of years of structure that was built up for those the fungi to be able to get into those areas. If anything, I could see fire inhibiting some of those, depending on how often you're burning, because although for those heat changes, they don't happen too far in the soil depth, but it's at the very top where you're having some of those communities be really productive.
Yeah, and especially with this prescribed burn, I don't think it's getting hot enough to really kill any of the microorganisms. And some of the data I showed show that really the amount of carbon utilization didn't change at all after the fire. So the microbes that are inhabiting that soil in either community are really driven by the site more than by the fire. But there may be different responses to fire as a result of these differences in communities. So there might be a stimulation of carbon utilization and CO2 production right after a fire in a restored prairie that doesn't exist in a remnant.
Okay. And I did get the specific question a little wrong. They were asking specifically if they were to add or be composting tea into the restored prairie. Would that help?
Paul, we're looking into it.
Okay.
Look, get back to us in 3 to 5 years.
Right.
Okay.
That's a great idea.
Okay. Another question we have here is, what implications might the altered microbial communities have, specifically on the prairie community? Reduced native diversity, increased, seems like you read this all, that all popping up, increased presence of invasives, or other implications?
I think it could be a decreased resilience to change is one thing. So, for example, as the climate continues to change and maybe we get warmer summers, we might have a different response in these systems that may be driven by microbial factors that that aren't quite as resilient in the restored prairie as in a remnant. Definitely a lack of arbuscular mycorrhizal fungi is going to inhibit and not promote as much above ground biomass growth as if they were there and robustly in the community.
Okay. And I believe we covered this, but I wanted to make sure that this question is still asked. Do you think fire is playing a role in shifting that microbial community toward remnant microbial community?
So some studies have shown that restored prairies do start to progress toward what looks like a remnant community, especially if fire is being reintroduced into the landscape. It's something that studies also show that never quite gets there. So you never quite get back to exactly what the remnant soil community looks like, or what the remnant soil community can do, at least from studies that have been published. We don't know that in our study yet, but that is also something we're looking into, using a technique that looks at really fine-scale functions of microbial community structure using molecular techniques. That's really the next step of where we're going, is looking at really looking at biodiversity and fine-scale functions in these systems and seeing how they're different.
Yeah, this is only year one.
Sure.
Well, September, actually,
Oh, yeah.
so it's 6 months, yeah.
Another question just came in. The question is, what about remnants that were farmed just once or twice? Do you think these will shift totally to the microbial that's typical of farmland, or back towards more of a remnant?
I guess it would be on the intensity of the tilling that occurred during that time. I'm not aware of anything that studies the occurrence of different farming practices and looking at year after year how those communities change. But as Kathryn mentioned earlier, when you have some pretty intense farming, they have some long-term implications for that.
Yeah, I think tilling is going to be an immediate effect on the influence of the fungal portion of the community, and then it just depends on how much and how long the nitrogen would have been added as to whether the bacterial community is really going to change.
Okay. Another question that just came in. It's asking, where can we find out more about your AMF methods?
Where can we find out more about the AMF methods? The first thing we're going to do, and we haven't done any AMF work yet, so the first thing we will do is an assay called phospholipid fatty acid analysis. So it tells you basically the proportion of AMF based on the amount of AMF lipids in a soil and the proportion of that to bacterial lipids. So that's a nice coarse way of measuring how much AMF is in this soil. And that's honestly what's often been done in literature that's looked at restoration with microbial communities. The other thing we want to do is start to look at, well, what about the diversity of the AMF that's present? So if there are different versions of AMF, different strains or species of AMF in the soil, is one better than the other? So we can do that using high throughput techniques. If this is a molecular person that I'm answering a question to, then yes, it is very tricky to do. And we're working with somebody at Argonne National Lab who is able to determine metagenomically and using high throughput sequencing what types of AMF are present.
Okay. Two follow-up questions here. Maybe we can do them together, and it's relating to the diversity. So you're looking at both the remnant and restored. What was the diversity of the remnant? Was it high diversity or low diversity starting out compared to the restored?
In terms of plants?
Yes.
Yeah, the remnant was, well, I mean, there was some basic assessments on the floral communities that were there, and I don't know how it's about. I don't have a number.
Well, so both of them were dominated by big blue stems. So about 75% of the community had big blue stem, at least our plots. But then with our data, we saw that the restored really had only 3 or 4 other types of species, and the remnant really had 11, I think. So
yeah, and I will just add on there that because we started this pretty late in the growing season, we did miss some of the diversity because we couldn't ID all of our forbs to species. So in the continuing years, we will be getting better diversity measures. But yeah, we had definitely higher diversity of forbs in our remnant.
From a prairie enthusiast perspective, it feels good to walk in there. How does that sound?
Okay. And related to it with the burning, it sounds like you were doing more of a dormant season. Are you going to be exploring to see if there's any difference in the season of burning and the effect on the microbial community, so a dormant versus a growing season burn?
Yeah, I think that, that's something that we're going to have to talk about as we, so far we've just looked at the 1993 planting in the remnant, and as we start to examine the chronosequence with the new planting and the 2010 planting, we will have to keep in mind that although we do want to look at different seasons, we have to see what's going to be best as we try to compare across the chronosequence. Would you agree with that, Kathryn?
Yeah, I agree. And again, it's also going to be something that is based on KNC and Ryan's schedule and, you know, the crew that Ryan has to do things. So we do have to consider, you know, experimentally what is also feasible.
Right. There's only so many layers of those variables you can start adding to make the study go along well. And, you know, taking those abiotic metrics too on temperatures of the fires, if we do have differences in seasonality, may at least give us some insight to what those differences might be.
Yeah. Typically, from a bacterial and below ground perspective, bacterial communities don't tend to shift that much seasonally, but the fungal communities can. So it may be more interesting from a fungal perspective to take to look at seasonality of the burn and
right. Short answer is we're still going to be doing mainly spring burns, early spring burns, or late fall burns. The conditions, it was kind of an anomaly day for that growing season burn, so jumped at it. But most of our burns are going to be in the spring, and then small percentage in the fall.
Okay. We do have one more question that just came in.
Okay.
It says you emphasize increases in nitrogen immediately after burning. Several studies indicate decreases in nitrogen. Is this related to time since the actual burn?
Yes, it really is. So immediately after a burn, you're getting all the surface nitrogen that hasn't leached into the ground yet. So we collected within 24 hours of burning. Many studies that look at nitrogen have been performed in, especially in forest fire systems. And in those systems, you're dealing with a much more intense burn, and usually people do not sample the day after. So we're getting a lot of surface nitrate and ammonium that remains in the system following the burn into October, as we were showing.
And these sites will be sampled seasonally as well, too.
Well, if we can get out there with our 4 feet of snow, we're going to try to go out in February and get a nice winter sample, but that may not be feasible. We're going to do it.
Metal detectors.
Yep.
Good. I guess one kind of just final follow-up, I mean, and this is looking at the manager scientist kind of lessons learned. You guys had excellent collaboration. It looks like you got some real good plans for the future. Kind of the key to that sounded like it was kind of that pre-proposal, figuring out your methodology in advance. Do you have any kind of last-minute suggestions for other people looking at doing collaboration on fire research and fire management?
I think regular meetings, and I think that Ashley's position is really important to this collaboration too. I don't think it would have happened unless Ashley had the academic researcher and land manager side to her job. So I think that's a really important position. Ashley, do you have any things you want to add?
No, I really think just, sometimes over coffee or over a beer, just talking about our interests with really open dialogue and being open to, the advantages of working with someone who's really different than you in their interests and figuring out how it works. And I feel like we should give shout-outs to our other collaborators that are far, far away. But, yeah. So we're building a really great team, and most of us all of us are really early in our career and really motivated. So I think that all of those kind of have combined to, for a really great project that I know I'm really excited about. And yeah, I'll leave it there.
I've found it very beneficial to have some like cross-discipline teachings too as we're going through this project. So I'm handing over a lot of technical knowledge, and Kathryn's handing me a lot of reminders on my my biology background. So it's really good to find someone that, although you have similar goals, that you're picking people that have really good strengths in their own areas, and not a whole bunch of people collaborating with very similar strengths. I find that diversity was helpful, as long as, of course, you're open in your thoughts and your willingness to learn too.
Yeah, I think one of the greatest moments of our collaboration was I asked Ryan for some restoration literature, and a week later he gets back an entire folder of something like 25 papers of his favorite papers about restoration. So it was really great to have somebody who understands the science and the land manager aspects and just say, here, read these, you'll understand, a lot of what you need to know, not everything, but it's been really helpful to have both of these guys helping out with teaching me about restoration and being open to thinking about microbial dynamics.
Yeah, one thing I didn't realize is there's a lot of the literature on burning in tallgrass prairies and response to them is pretty much taking place all out west, and there's very little literature about eastern tallgrass prairies in regards to response and timing and all that stuff. So
Yeah.
that was something I picked up on too while going through this.
All right. Well, this has been great, guys. I'd like to thank you all, Kathryn, Ryan, and Ashley, and everyone online for joining us this afternoon for a very interesting webinar and discussion. If you guys could just bear with us for just a few more minutes, we have a few announcements. First, if you would like to review the webinar or share with someone that might be interested but missed it, we're going to have a recording of this webinar that will be available on the Lake States Fire Science Consortium website. Should have that up tomorrow, and I'm sure Craig will have it up on the Tallgrass Prairie Oak Savannah website also. And then we want to take the time or opportunity here to highlight 3 of our upcoming webinars. The next one is also a co-sponsored webinar between the Lake States and Tallgrass Prairie and Oak Savannah Fire Science Consortium. That's going to be on February 13th at 2 p.m. Eastern, 1 p.m. Central. We're going to be looking at the multi-scale responses of eastern Massasauga rattlesnakes to prescribed fire, and that'll be presented by Matt Cross. Greg, are you on?
Yeah, thanks, Jack.
Okay.
And then the next webinar, in addition to the co-sponsored one, is coming up for us on February 11th, and that's going to be a little bit earlier, noon Central and 1 o'clock Eastern. And the title of that webinar is An Adaptive Framework for Management of Invasive Forest Plants, Integrating Prescribed Fire, Mechanical, Chemical, and Biological Techniques. The presenter will be Sean Blomquist, an ecologist with the U.S. Fish and Wildlife Service at the Ottawa National Wildlife Refuge in Ohio. This one will be emphasizing more woodland communities, including oak savannas, oak hickory type forests, and floodplain forests.
And then the next regularly scheduled Lake States Fire Science Consortium webinar, that's going to be February 20th, again at 2 p.m. Eastern, 1 p.m. Central. And it'll be assessing the drivers of the spring dip in foliar moisture content and their potential impact on forest fire behavior. It's going to be based off of a lot of red pine and jack pine needle samples that were taken in Wisconsin sites last year, and how there might be ways to predict a little bit when that spring dip season is coming on. In order to prevent some of the large wildfires. That'll be presented by Matt Jolly. He's a research ecologist with the U.S. Forest Service. Finally, before I shut down the webinar here, when we shut this down, there should be a survey box that'll pop up. If you don't see a survey that pops up, it might be because your browser's blocking it, so you could go in and take a look. If not, give me an email. But we would appreciate if you guys could take the survey, because this survey information is very important to help us so that we can better serve your fire science needs. So this concludes today's webinar. Thanks again to Kathryn, Ryan, and Ashley, and everyone for participating today. I want you all to have a great rest of the day. And Kathryn, Ryan, and Ashley, if you could just stay on the line just a minute. Thanks.